My name is Songwe Fanuel. I come from Zimbabwe, and I work for the Midland State University. So today I'm going to be talking about a technology or a tool that has been in existence for only 4 months, but the potential in which it holds needs to be investigated. All right. Let me start by quoting the 2018 Nobel laureate chemistry. In her Nobel acceptance speech, she said, let's see. Okay. She said that today we can, for all practical purposes, read, write, edit any sequence of DNA, but we cannot compose it. She was very confident when she was giving her speech that we cannot compose any sequence of DNA, but we can manipulate it somehow. We can read, write, edit, maybe using CRISPR and other techniques. She went on to say, maybe we can cut and paste pieces from nature's composition that has been from nature's composition, but we don't know how to write the bars of a single enzymatic passage. However, evolution does. That's what she said. So in a speech, she was saying, back then, I think it was 6-7 years ago, she was saying, there's no way on this earth that we can create a functional protein from scratch. That's what she said, okay? Then in November last year, entered the ChatGTP for genomes. And it's called the Evolutionary Optimization, or EVO2. Okay? I think we all know the capability of ChatGTP and how it has changed the way we do research, the way we do writing. I think in the afternoon I was talking with Professor Ben when we were having lunch. He was saying modern students are very lazy because they are just manipulating this ChatGTP. They can't write anything and so forth. Okay? So that shows the extent to which this tool has changed the academia completely. Okay? Now, we are now talking of this evolutionary optimization tool. It's called the EVO2. In other words, let's call it the ChatGTP of genomes, okay? Just imagine all the tasks that ChatGTP can perform. This one can do for genome, and even better, as we shall see. And by the way, it was published in November in a preprint. I think the DOI for the paper is there. Okay, so let's look at, let us investigate what EVO2 is all about, and maybe briefly look at the paper. The paper was titled Genome Modeling and Design Across All Domains of Life with EVO2, okay? In the context of this paper, what is genome modeling and design? We all know that a genome is information that is required to build and run an organism. In other words, this whole genomic makeup of a cell. Then modeling, in the sense of that paper, means that creating a computer program that can predict how changes in the genome will affect an organism. Okay? And again, design, looking at this paper, they meant that using the model, this EVO2, to create new genomes or edit existing ones to make organisms that do useful work, including the gene drives. Okay, so this tool, or this EVO2, was made by these 5 organizations, but at the center of this was the Arc Institute, assisted by NVIDIA, University of California, Berkeley, and USF, and Stanford University. Okay, so basically it represents the largest AI model in biology to date, and it is capable of predicting functional impacts of genetic variation, as well as designing novel genomic sequences. In other words, new proteins that are not as a result of evolution, contradicting that Nobel laureate in 2018. So with this one, we are able to come up with new genes that have never been in existence from scratch. So Evo2 was built by, it's built on NVIDIA's DGX cloud platform, and it was trained on 9.3 trillion nucleotides. And these nucleotides, they came from 128,000 genomes across all the trees of life. That is from prokaryotes, mammalia, plants, and so forth. But 128,000 genomes is something that is huge. If we were to task ChatGTP to do that, we are instructing ChatGTP to read all the books and papers that are found in the world. Okay, so they trained this model using 128 genomes. That's like 9.3 trillion nucleotides. So the most intriguing part of this tool is that it can process, or you can input up to 1 million nucleotides into the search box, allowing it to understand the relationship between even distant genomes, the relationship between genes from, let's say, from a worm and maybe genes from a human, and it will just process and give you the relationships. 1 million nucleotides. It's equivalent to a full genome of some bacteria, or even half there. Okay. The other thing is that it can predict the effects of both coding and non-coding mutations. Okay, so it can predict the mutation within the exons and the introns. That's the beauty of this thing here. And there's the picture of the platform where you can do the prediction, the manipulation, and so forth. Okay. Okay. So how was it built? When they were training the model, the validation of Evo2 capability shows that it can identify how mutations can affect a protein, RNA, and even the organism's fitness. Okay. So in that paper, they actually asked the model using the BRCA1 gene. The BRCA1 gene is one of the most studied genes in cancer biology. Okay, so most of the regions of that genes are known, the mutations are known. So they sort of tested the machine using the BRCA1 gene, which they knew. And interestingly, it achieved more than 90% accuracy in predicting all the mutations in that gene, including the benign and the disease-causing regions. So why should we be concerned, or why is this tool so special that it deserves to be spoken about, like what we are doing? Number one, like I said, that it works on all domains of life. Like you can put in bacterial genome, plant genomes, and even human genomes. Okay. It uses evolutionary principles in contrast to what Arnold, the lady, said. So in other words, it learns from nature that has already designed genomes over millions and millions of years to design new genes. And one thing is that it is super accurate, such that it can predict even small changes in DNA that will affect an organism. For example, if you change one gene, will the organism grow faster or produce a new chemical, or maybe stop working altogether? Okay. Evo2 can even suggest new ways to edit or create new genes or genomes for you. It can even suggest for you. Okay. So when we apply this thing in biogenetic control, it can lead to groundbreaking advancement, but it can also raise ethical and practical issues, safety issues that were being talked about. Okay. So let's try to evaluate this Evo2 using this movie here. It's a 1966 Western movie. I think one of the best Western movie to be produced ever. It's called The Good, the Bad, and the Ugly. I think some of us might know the movie. It was made up of 3 characters. There is Blondie, the good. Is cunning and morally ambiguous bounty hunter. Then there is Angel Eyes, the one in black, I think, a very ruthless mercenary. He was only loyal to money. You give him money, he will do whatever job you ask him to do. Then there was a Tuko, an ugly bandit, who was very opportunistic and very cruel too. But however, their paths crossed, they seek each other treasure, leading to shifting of alliances and so forth. So let's look at evil 2 from the good, Blondie. The good, its application in biogenetic control. Okay? We can use these two to design synthetic genes. Okay? So this model can design new genetic sequences that never existed in nature, potentially creating synthetic genes that could be introduced into pest populations to control their reproduction and survival. We continue the good. It can help us understand the functions of a gene, okay? So we can just get a chunk of DNA, put it in there, ask it what this gene is, what can it do that it doesn't doing, or even introducing a mutation into that gene, what will happen, it will tell you. It can also predict pathogenetic mutation. It can identify disease-causing mutations in humans. It also has the ability to predict harmful mutations in pests, aiding in the development of targeted biocontrol strategies. Okay? Then one interesting thing is that it can analyze and predict outcomes much faster than traditional lab experiments, saving money and time. In one of the experiments, one paper that was published in January, scientists used this tool to create completely new antivenom proteins. They said something that was very shocking because they predicted the antivenom for, they predicted using this tool some proteins that we can use against snake venom. And when they tested the proteins in the lab, they worked at one go. So there's no trial and error, and so forth. And the paper is published in January. It was called De Novo Designed Protein Neutralizing Snake Venom Toxins. Again, the other good is we need EVO for agriculture and food in Africa. It's actually good for us. Let's look at EVO, the bird, the angel eyes, complexity and accessibility. It's a highly advanced tool that requires expertise in bioinformatics, meaning that it might not be accessible to smaller labs in developing countries. It's a problem. We saw this during the COVID era, where most African countries were failing to create messenger RNA vaccines. So it's just a preserve of those European countries. Ethical concerns. 1 minute. Okay, let me round up. Ethical concerns, I think we've talked about some of these things. Costs and the resources. Then let's look at evil, the ugly. Dual use technology, like what Labi said yesterday in his lecture, that AI lowers barriers for non-experts to develop threats. So they can be seated in their garage, creating very pathogenic microorganisms, some that are even resistant to antibiotics. So can be used for harmful purposes. Loss of biodiversity. If we start designing organisms from scratch for specific purposes, it could lead to loss of biodiversity. Inequality and misuse. Unpredictable long-term effects. Then looking ahead, I'm sorry because I was thinking the presentation was going to be 20 minutes. Let me just wrap up. Okay, so the creators of this tool, the Arc Institute, said they envision EVO2 as an operating system, a foundation layer that provides a platform for broad generative functional genomics. So anyone can build their app upon this thing, and scientists will keep improving EVO2 to make it even more smarter, like designing the entire ecosystem. Like what the Arc Institute is saying, they want to design a virtual cell So just as good as we had ChatGTP, then in came DeepSeek. That is more powerful than ChatGTP. All right. So I was going to wrap up my speech on how to harmonize or how to regulate some of these technologies, but unfortunately my time is up. Thank you very much.